Thank you, Stefan. I really appreciate it. And thank you for everyone that are here this afternoon. So I'm going to talk about reverse genetics approach that we have used over the last 20 years in terms of vaccine design for Africa, and especially in our region in Southern Africa. So Southern Africa is mostly free of FMD, although we have it endemic in the northeast corner, and around that area where FMD is present in wildlife, we have a vaccination zone. And basically our control strategy is creating different layers of barriers, and the barriers consist of a physical barrier as well as a immune barrier. Now, the importance of wildlife, and specifically buffalo, is that buffalo inside this park in the northeast corner of South Africa, the Kruger National Park, with times escape this physical barrier. And due to high cattle densities in this area next to the park, we have to vaccinate annually. And it's about 300,000 cattle that we have to vaccinate. However, we have 3 SAT serotypes, or 3 serotypes of FMDV, and that is the SAT serotype, SAT 1, 2, and 3, that we have to create an immune barrier for in order to make the vaccines effective. Now, we have done studies, and we have presented it before, that in the Southern Africa region, when you make vaccines from a geographical region, it may not protect viruses from another geographical region. So our approach in terms of vaccination and to improve vaccination is to make use of reverse genetics. And basically the definition of the reverse genetics is that you convert the full RNA genome of the virus into a cDNA copy, which you clone into a plasmid, and then you can produce RNA from this DNA in vitro, or you can use the plasmid itself to transfect into BHK cells, after which you get transcription and translation, and the proteins of the virus will assemble itself to make new virus particles. So now you can introduce mutations in the genome to have several effects or improve the virus characteristics and so on. So in one experiment where we swap the capsid coding region of a SAT2 virus into the SAT2 backbone that we created in this manner, we created an intraserotype virus, a SAT2 chimera, and we made vaccines of these two and vaccinate 7 animals with each of these. Now, from the clinical scores, you can see that these animals were all protected compared to the placebo controls, which had severe lesions and systemic spread. So the chimera react very similar to the wildlife virus. So now one can use this genome length clone or chimera clone and make some improvements. And over the years, we have also identified the amino acids that are involved in the adaptation of the virus on cell cultures like BHK cells that are used for production process. Generally, this adaptation process involves certain amino acid changes. We have characterized those, and generally there is positive charges around the 5-fold axis that accumulates, and these positive charges that accumulate on the 5-fold axis allow for the binding of a heparin molecule when you do some modeling. When you look at the character of the virus, which have these positive charges, it also grow on CHOK1 cells, which do not generally have the alpha V receptors that FMD used to enter, so it must enter by alternative receptors. When we want to introduce these positive charges to a virus, typically a SAT2 chimera, and you grow this in suspension, you actually can improve the titers by such a chimera where the original wild type virus did not grow on these suspension BHKs at all. But the question is always, when you introduce mutations to it, does it also have antigenic effects? We might used in this case another virus, SAT1, SAT2 chimera, interserotype, and we compared a vaccine made from the interserotype chimera virus with that of the wild type SAT1 virus, and the interserotype chimera virus has the positive charges on the 5-fold axis. The immune responses, antibody responses after 28 days of vaccinations were comparable. And when we did some challenge studies, the PD50s, so this was full dose, quarter dose, and 16 dose for both vaccines, the PD50s were very similar. Although 2 of the chimera vaccinated animals did show clinical lesions on one of the hoofs. So there was some systemic spread in some of the animals. When we work out the PD50s, they were comparable. So it was acceptable, within acceptable range. So the other aspect that we worked with was about the stability of the capsid and improving the stability of the capsid. And this is work Brian has presented a bit of it in relation to the empty particles vaccines that they do. We have done this work with Plum Island, Pirbright, and Oxford University in MSD Animal Health at that point. And so amino acids at the interface of these of the capsid have been identified by the Oxford group that we could mutate, and we made these mutations, and we did found some of these mutants where the stability of the virus was actually improved. These was not cysteine residues like in the case of the empty particles, but it was increased hydrophobic changes. We made vaccines of these, and we compared the stability vaccine with the wild-type vaccine. We followed these animals over 160 days, and Mariano already gave a quick overview earlier today about that. And we got full protection. We also included another stability mutant as an antigen that also provided full protection. When one look at the antibody titers, and this is basically some of the information that Mariano showed, but what was important for us was that once we did a single vaccination, when we look at the total antibodies, this is the liquid phase and this is the solid phase competition ELISA. When we look at total antibodies, that antibodies declined very quickly. And we had to follow up with a booster vaccination about 2 weeks after in order to have the antibodies increased again and have these antibodies for a long enough time to make this vaccine at all usable. Also, neutralizing antibodies, although the initial drop in the neutralizing antibodies was not so dramatic, with the 2 vaccinations, we had good antibody responses for up to about 6 months after the initial vaccination. But around 5 months after the initial vaccination, there was a decline, and we had to make the decision to do the challenge. So we did the IgG1 and IgG2 as well, and there was increase in these isotypes similar to the neutralization antibody profile. And if I can go to the next one. I think I pushed it now too many times. Okay, so basically, we have some success stories with reverse genetics, and I just want to summarize it. We have made a total of almost 80 different mutants that include the cell adaptation changes, the engineering, the stability. Some of them did not work, some of them was not even viable. And we were able to do at least 5 different animal trials using different of these constructs. So one of the future things that we and we have started to explore that is to combine these mutations into capsids, both cell adaptation and stability. But there's another aspect that we also wanted to look at, and that was, is there any possibility to improve the antigenicity? We talked a lot about vaccine matching this morning. Is it possible to have a vaccine that have a broader spectrum of reactivity within a serotype? It might not be possible to have universal vaccine for across all serotypes, but within a serotype, that you have better and broader protection. So we gathered also over the years a number of information about epitopes and antigenic regions for the SAT viruses on the capsid. Generally, it's surface exposed regions that are exposed. These regions we have identified by means of sequence alignment, as well as neutralization titers, combining those. Richard has presented in the past on that. We've also used single-chain variable fragments, which is basically monoclonal and very small monoclonal antibodies to identify these regions. And we have made mutations in the SAT2 genome length clone, changing these antigenic site or potential antigenic site to that of a donor virus that was a little bit different. We did the VNTs studies.
2 minutes.
2 minutes, with both the serum from the epitope donor and the original. We got increases in neutralization profiles, and those increases in neutralization profiles we could link to increase in avidity as well. And then we used monoclonal antibodies to find a profile or reactivity and try to identify which of these amino acid sites that we changed actually work together in the interaction of a monoclonal antibody. And there was one site that was quite important in the VP2, we call it VP2-2A, based on the O antigenic sites, which completely abolish monoclonal antibody binding. And then there was profiles that also include another site in VP2 and a site in VP1. And we had the 5th monoclonal antibody that had a complete different profile. We went further to this with this work. We made alanine knockouts of epitopes. We also swapped epitopes to that of a completely different virus that's not even in our region. Again, we linked increase in neutralization titers with increases in avidity. And the monoclonal antibody profiles was very similar. Again, these 4 monoclonals, those and the DA10 gave similar profile. This site in 2A abolished the binding, and the other sites in VP1 and the other site in VP2 decreases the interaction. And this monoclonal have a different profile. The epitope swaps to that of the Egypt virus also made sense in terms of that the VP2 between these viruses were the same, so there is no swap to a VP2 Egypt epitope because it was exactly the same than our virus. And we can map, using these profiles, an area where these monoclonal antibodies bind, which has been confirmed by structure. And the big thing that I want to use to conclude with is that we can use this reverse genetics and combine it with conventional inactivation for vaccines. And we are also able to do changes in the epitopes that increase avidity. However, these last ones we haven't ever tested in animals. I wanted to thank people in my lab and people we collaborated over the years. Thank you so much.